All right, so what we're going to do here is we're going to, and I missed a slide here, so let me get back up here. There we go. Okay, so what we're going to do here is talk about gene expression, and this is chapter 18. And so what we're going to do is we're not going to go over as much detail maybe on the next few chapters. There's just some concepts we want to go over real quickly. And again, this is more cell molecular class or bio 8 class, but I do want you to have some concepts here. And so this first one is looking at how genes are expressed. So in the last one, we talked about how to make these proteins. But the question now is what decides when they're made, when they're not made, when do we actually turn these genes on or off? Because every cell in your body actually has all the information necessary. So the question is, when does it become important? So here we got a picture of this fish. You can see it up on the top. And in the eyes, the way they swim, they're right at the surface. So some of those retinal cells on top need to be able to see and optimize in the air. And then there are some that need to be better at seeing underwater. And it really comes down to they're expressed differently due to their specific functions. And that really comes down to, some of these other parts that I kind of glossed over, like transcription factors, whether they're there or not, what gene actually gets read. Okay. And so it turns out, like for this one, they're showing you gene 3 is necessary in any eye. But then gene 1 and 2 will determine when is it best turned on and what that will do. And so certain cells in the eye that are going to be above water, maybe it's gene 1 that's expressed because of those transcription factors. And then maybe in the ones underwater is going to be gene 2. So this becomes real important in embryology and also when we're looking at what's actually for function. So one of the things we've decided in this class is you should probably have a basic understanding of operons. These are found really in bacteria, and we assume they work very similar in us, although we're finding out the ones in us are probably a little more complicated, but it gives you kind of a general idea. So an operon is this clustered gene with one promoter. It's kind of like an on-off switch, which is called an operator. And then there are ways of repressing it, shut it down. Those are called repressors. And then there are molecules that help the repressors, which are called the co-repressors. So what we're going to look at is two different types of operons. We have repressible operons, ones that transcription is inhibited, and then we have inducible operons where the operon actually stimulates transcription. And so we're going to give you an example of each. So we'll start with the repressible operon, and that would be the trip operon. And this is actually the one associated with tryptophan. And so tryptophan is an amino acid that you can actually make in your body. But it's also one you get in your diet. And tryptophan is known for being in turkey, and supposedly that was, it's one of the amino acids that make you sleepy. And so people thought that, on like Thanksgiving, where people eat a lot of turkey, if you eat too much, you get sleepy. No, you're getting sleepy because you eat too much. It's not the tryptophan. You cannot eat enough turkey to make you fall asleep. So anyway, so here's what you need. What you have to understand, this is a repressible operon. So here's what you need. When there's no tryptophan, the repressor is inactive and your operon is on. So if you don't have tryptophan, if you're not eating turkey and putting it in your body's running out of it, basically this operon is on, the repressors are not working, and you're making tryptophan. Okay, once there's tryptophan in your body, the tryptophan is actually the co-repressor. It actually hooks to the repressor, makes it active, slides it into the operon, and turns it off. So that way you're not making more tryptophan because you already have enough. So when the tryptophan starts to break down or it gets used into other things and you're low on tryptophan again, they will slide out of the repressors become inactive, and then your body will make more tryptophan. So that is a repressible operon. The other example is the inducible operons. And inducible operons are the ones that something has to be there to actually make these things run. So here is lac operon is talking about basically lactase. And so what happens is lactose is absent. You don't need lactase. So the repressor is active, it is sitting in there, and it is doing what it needs to do, shuts it down. Okay, so you start drinking milk. All of a sudden lactose is present, the repressor then becomes inactive. You have an inducer, and that inducer is allolactose, which is in milk, and that hooks on to the repressor, inactivates it, and now all of a sudden your body is making enzymes for using the lactose. Okay, so this is called inducible. So this is the opposite of what you want. So if I were you, if you were smart, you're not, but if you're smart, what you do is make sure you understand these lines on the bottom of each of these slides, well, what's actually happening. I think that's the easiest kind. I will tell you when you get especially a multiple choice question on these, a little hard sometimes to kind of walk through the steps, but if you have that bottom part memorized, that will definitely help you. Okay. So again, these are inducers. All right, and that's kind of there's also a positive gene regulation, and here we're using that second messenger camp. And so what happens is when you get a lot of, when you don't have a lot of glucose, camp is made. So you have high levels. And because of the camp, you're making a lot of messenger RNA for LAC and that promoter and operator. And so what happens then is it's trying to increase the amount of messenger RNA, so you make a lot of these proteins. And then when glucose is present, the campus levels are low, and then you don't make as much messenger RNA. So positive gene regulation basically means that when you have something, it's gonna help build even more of that, and that's really what we're. Negative is basically when something builds up, it starts to shut it down. This is the opposite. When something is building up, it actually increases something, and in this case, it's increasing the production of LAC mRNA. All right, so with that, we'll stop it. But we'll stop the share. But what we're trying to get at here is I want to make sure you understand this a lot more complicated than just reading and making proteins. Lots of little control things that we're not as sure about, and we're making strides in trying to figure out exactly